Ladies and gentlemen, good afternoon. My name is Nadia Islam, and it is both an honor and a privilege to stand before you today at the 7th International Conference on Eurasian Biochem 2024. I would like to extend our deepest gratitude to the conference organizers for providing us with the invaluable opportunity to present our research. Together with my esteemed colleague Betül Akçeşme, we're delighted to share our paper titled Investigation of Phytochemicals as Potential Therapeutics for Multiple Sclerosis by Computational Approaches. I'm presenting on behalf of both of us, and we sincerely thank you for this platform to contribute to the ongoing scientific dialogue in this critical field. Thank you very much. In this presentation today, I will be talking about phytochemicals as potential therapeutics. We will be discussing the methods in silico screening, Dr. Duke's database, Campbell database, and some protein targets. And finally, we will be talking about future prospect and conclusion. Multiple sclerosis is a chronic autoimmune disorder characterized by myelin degeneration in the central nervous system, leading to various neurological symptoms. The pathophysiology starts by immune system attack when the immune system mistakenly attacks the myelin sheath, the protective covering of nerve fibers. That leads to inflammation and subsequent damage to nerve fibers, disrupting communication between the brain and the body. The common signs and symptoms includes vision problems, emotion and cognitive changes, physiological problems. The current treatments have a lot of limitations. We have DMTs and we have symptomatic treatments. We have FDA-approved DMTs, approximately 22 of them, including interferon beta, glatiramer acetate, or dimethyl fumarate. But up until now, there is no cure for MS, and there are potential side effects, including flu-like symptoms, fatigue, increased risk of infection. Phytochemicals are alternative to current treatment. They're naturally occurring compounds in plants. They offer potential therapeutic benefits, serving as promising alternatives for MS treatments due to their anti-inflammatory and neuroprotective properties. We have 5 different classes of phytochemicals, including flavonoids, alkaloids, terpenoids, saponins, and polyphenols. Aim of the project is to identify those phytochemicals from the literature then associated with MS, from Dr. Duke's database, then from Campbell. We will then analyze protein targets of FDA-approved drug to gain insight into the mechanism of action, and finally to explore new drug target proteins for identified phytochemicals, including using the omic data. In the first part of the study, we used an in silico screening process. In the first step, we did some data research. In the second step, we used Dr. Duke's phytochemical database, finally Campbell database. The selection criteria for the phytochemicals were biological activity and efficacy in modulating immune response, potential to reduce neuroinflammation and protect the myelin sheath, safety profile, minimal adverse effects. Of course, applied rule of 5 for drug likeness assessment. From Dr. Duke's phytochemical database, we identified 1,708 chemicals associated with MS, and these 5, they had the highest activity count. Activity count in this database represents the number of biological or pharmacological activities those have been documented for a specific phytochemical. This count reflects the breadth of research and evidence supporting the various effects that one particular compound may have on health and disease. We have caffeic acid with 18 count, we have quercetin and curcumin with 16, we have apigenin 12, I beg your pardon, apigenin with 14 and ascorbic acid with 12. In this slide, we can see the sources of these phytochemicals. Caffeic acid, as the name suggests, usually found in coffee and various kinds of fruits. Quercetin, usually found in onions, apples, berries. We have curcumin, naturally found in turmeric. We have apigenin, found in parsley, celery, chamomile tea or flower. We have ascorbic acid or vitamin C, usually found in citrus fruit. Acts as potent antioxidant, regenerates other antioxidants. For caffeic acid, it usually acts as an antioxidant by scavenging free radicals, inhibiting enzymes involved in the inflammatory pathways. We have quercetin. It inhibits histamine release, modulating key signaling pathways involved in inflammation. We have curcumin. It inhibits nuclear factor kappa B. That results in downregulation of pro-inflammatory cytokines enzyme like COX-2. We have apigenin. It's an anti-inflammatory, anti-cancer component. It inhibits enzymes such as COX-2, modulates pathways involved in cell cycle regulation and apoptosis. Finally, we have ascorbic acid or vitamin C. It acts as an antioxidant by neutralizing free radicals. It regenerates other antioxidants, supports immune system, enhancing the activity of phagocytes and T cells. In the second part, we will be talking about drug target proteins. From the literature, we found SIPR, IL2R, CD25, alpha-4 integrin, B-cell CD20 antigen, PNP, DHODH, FAEs, type 1 interferon receptor, glucocorticoid receptors as potential drug targets. So the S1PR is a critical component which regulates a lymphocyte that engraves from lymphoid tissues. We have IL2R CD25. It's essential for the activation and proliferation of T cells. We have alpha-4 integrin, involves in adhesion and migration of immune cells across the blood-brain barrier. We have B cell CD20. It's an antigen expressed on the surface of B cells. PNP is an enzyme crucial for proliferation of lymphocytes. We have DHODH, which is an enzyme involved in the de novo synthesis of pyrimidine, which is required for DNA and RNA synthesis, as we know. We have FAEs or dimethyl fumarate is to modulate immune response, activate nuclear factor erythroid 2 related factor 2 or NRF2 pathway. We have type 1 interferon receptors targeted by drugs like interferon beta. We have glucocorticoid receptor mediates the effect of corticosteroids, which are potent anti-inflammatory agents. In this slide, we can see Dr. Duke's 5 phytochemicals versus the targets. Caffeic acid has anti-inflammatory properties. This can interact with cytokines like IL2R, CD25. It's not well established, but there are literature. We have quercetin has been studied for anti-inflammatory and immunomodulatory effects, maybe indirectly influence S1P or signaling pathways. We have curcumin, and it has this anti-inflammatory immunomodulatory properties as well, can bind to type 1 interferon receptors. Apigenin and ascorbic acid, with the current literature, we could not find anything right now, but the research is ongoing. Finally, Campbell database. We followed a very strict method, such criteria. We use multiple sclerosis, small molecules, phase 3, RF5 violation 0, RF5 violation certain molecular weight. And then we found lakinamide, which is an oral immunomodulatory drug, and we found 20 proteins associated with lakinamide. So if we can wrap up this presentation today, we can say our key findings were effective phytochemicals identified from the literature. There were a certain number of them. We had 5 top active phytochemicals from Dr. Duke's database, and we had some 31 related proteins from 9 from literature research and 22 from Campbell as promising drug targets. This is an ongoing investigation, so The binding and interacting analysis of potential phytochemicals with target protein will be investigated using in silico molecular docking tools. We will be running some molecular dynamics simulation, and this will be validated for in vivo and in vitro studies. And in conclusion, we can say this database has identified effective phytochemicals promising drug targets for MS therapy. The integration of these compounds open new avenues for drug development. Targeting 31 identified proteins provides a robust foundation for future research, advancing our understanding and treatment of MS. These are the references we used for this study, and thank you very much for your attention. If you have any question, feel free to email us. Thank you very much.